Kidney organ culture and organoids that are derived from programmed stem cells provide excellent ways to study mechanisms behind kidney development and disease. The current organ culture system do not provide blood flow to the kidney, however. Due to this reason, the current models are incomplete and fail to recapitulate the main function of the kidney, which is blood filtration. In this publication, we present an improved protocol for cultivation and visualization of embryonic kidneys and organoids on the chorioallantoic membrane of chicken embryo. In our novel experimental setup, we overlay the kidney rudiments transplanted on cam with permeable mini reservoirs that are filled with culture medium. This protocol increases significantly the survival of kidney rudiments and also their intrinsic endothelial cells. The method enables blood flow to the developing glomerulus. Recently, we published also a novel fixed Z-direction organ culture method that offers high-resolution confocal 3D time-lapse imaging. The method provided optimal conditions for long-term confocal imaging of the organoids and organ rudiments. In the approach, we gently compress the tissue between a glass cover slip and a permeable membrane. Here we show how the custom designed plates are made and how the culture experiments are set up. Thus, in this publication, we present an improved renal organotypic culture technique. This method offers a better way to model organogenesis and conduct high-resolution imaging to study embryonic kidneys and renal organoids ex vivo.